in the opening cartoon of this last lecture of this section of the class, Calvin reads in his math textbook, I guess, a bushel is a unit of weight equal to 4 pecks. And then he asks Hobbes, the tiger, what's a peck? And the tiger goes, it's a quick smooch. And then the reason I put this up is I can just visualize some of you Wednesday night studying for the midterm going, oh, you know, I don't understand math at all. I'm hopeful that that's not a preview of you on the midterm. Biology is terminology laden, and some words have highly specialized meanings. So for example, I will disclose one question on the midterm to reward you for having come. There will be a question about the word antiparallel. One of your classmates named Andrea and I were having both a real-time and then an electronic dialogue about next generation things in molecular genetics that connect to medicine. And one of the reasons I love teaching is I get to keep up with the literature better than I normally have time for due to my full-time job. And in that conversation with Andrea, we came across this pretty remarkable article. It actually just came out 4 days ago. The PubMed ID is here, and the link to an article I recommend you read is here. Now, while this won't be on the midterm, in fact, it probably will not be even in any of the upper division classes that you're going to take here at Berkeley, save perhaps MCB 140, which is genetics for people who love pipetting. It brings completely new meaning to the real reason I started with Calvin and Hobbes. This is the original Hobbes, the philosopher and political scientist, the author of Leviathan, who was the first one, to the best of my knowledge, to coin the Latin phrase nos se te ipsum. Any classics minors? What does that mean? Who said that? Good for you. Know yourself. Well, this is Mike Snyder. He's a professor of genetics at Stanford. And this paper that came out brings new meanings to nos se te ipsum like I have never seen before. Next generation technologies that were developed literally over the past 5 years, so when you were in high school, they didn't exist yet, allow one to sequence the complete genome of a human being for less than $20,000 and in less than a month. Perhaps more remarkably, these very same technologies allow us to evaluate the gene expression program of an individual, not gene by gene. So is lactase on, or is this other gene off? But all of them, all 33,000 or so gene units in the human genome can be studied with respect to their expression level in real time. So what Mike Snyder did is he basically had himself monitored using all of these pretty remarkable next generation methods. So his complete genome was available. He was sequencing his blood's peripheral blood mononuclear cells, and he did that for 14 continuous months to produce what he calls an IPOP, which is an integrated personal omics profile. And what did that produce? Well, that produced one of those classic pictures from next generation papers, which is what my 14-year-old daughter would call TMI, too much information. So these are obviously the chromosomes in the human karyotype. There will be a question on the midterm about the number of chromosomes in the human genome. And that's the other reason I put this here. But so not only do we now know about Mike Snyder every single genetic variant that his genome contains, but we know over the course of about 14 months which genes went up in expression, which genes went down, how he differs in that regard from the average human being. He had 2 infections over the course of those 14 months. How his body responded to infection was tracked down. Most remarkably, and this is the real reason I'm teaching you this, is it turned out that he's developing diabetes, and his genome and these next generation analyses of his gene expression program predicted the fact that he's going to get diabetes before any available medical test could. So this is one person, one incredibly accomplished and well-connected person with access to completely cutting-edge technology. By the time, I was just talking to a student, a classmate of yours, who wants to be an obstetrician. So by the time she has her own practice and is board certified, it will be, in fact, probably quite feasible to take a parent, an expectant mother, and say, you know, Mrs. Kim, would you be interested in having your IPOP so that we can track you through the course of pregnancy and track your baby through the course of pregnancy to see how things are going and to maybe adjust your diet a little bit, adjust your exercise regimen. Folks, this will happen. In other words, it's not a question of will this happen. It's binarily the case that this will happen. The question just is with what kinetic. And so the reason I, of course, brought this up on the Wednesday before the Friday of the midterm is to highlight to you that the midterm will come and go. It's just a 50-minute experience, and 10 years from now, you will not remember that midterm at all. But what I'm passionately hopeful you will remember is what you had to study for it. That is, for example, things like the regulation of gene expression. And a good place to start, by way of trying to understand where we're going with this, is to read this news article in Science, on the website of Science, and then pull down this paper using this PubMed ID, and just read the introduction and the discussion. The materials and methods I wouldn't read, there's just way too much technical stuff. Okay. Today's lecture will consist of two components. One will be a re-review of material that I'm hopeful you know for the midterm. I had a lot of topics to pick for to re-review. I decided to pick what you're about to hear because, again, I think that when, you know, Mrs. Patel shows up and says, you know, my father died of heart attack, I'm about to have a baby, should I be eating more, should I be having more vitamins? And you need to be able to talk to Mrs. Patel about this in 2015 language, not 1955 language. And so that's why what's happening is happening. Okay. On Friday, I will ask you a few questions about this figure, as promised. And this figure is abridged beyond legibility, but I actually went through a much larger version of this figure, I think 2 lectures ago. And it shows you that the DNA to RNA to protein process is not academic or esoteric. It's very real world. The changes in the primary sequence of DNA produce changes in the primary sequence of messenger RNA, produce changes in the sequence of protein, and that has consequences on phenotype. And what I'd like to review and also drill down into the foundational layers of your prefrontal cortex is the notion of the relationship between the kind of mutation and the kind of effect that mutation can have on phenotype. I've said this to you before, and I'm saying this to you again because you will encounter this in your life. Once upon a time, there was a boy in Pakistan who experienced no pain, and he died. He jumped off a roof, and he crashed to death. We know why he had that property. He had that property because he was homozygous recessive for a nonsense allele of the gene encoding a pain receptor that is expressed in his spinal cord, specifically in the dorsal root ganglia. A nonsense allele is one which causes a premature termination of translation. So the messenger RNA is produced, just the ribosome is incapable of translating the full-length protein. Why is it incapable of doing so? Because it sees a stop signal. And that stop signal is there because there has been a single base change in the DNA encoding that protein. The consequence of that mutation on the function of the protein is pretty drastic. The protein no longer functions because the protein pretty much isn't there. The parents of this boy could sense pain perfectly well, but they were clearly heterozygous. They were carriers for this mutation. Why they were asymptomatic? Well, because the allele they passed on to their son is recessive. Why is that allele recessive to the wild-type allele? Well, because the wild-type allele does something. It lives in the dorsal root ganglia and transmits pain. And the recessive allele is loss of function. It doesn't do anything. This is why the wild-type allele is dominant over the recessive one. And a number of you are yawning, half because you're sleepy, half of you because this is the third time you're hearing this. It's about to get somewhat more entertainingly convoluted. And you're students at Cal. The top public university in the world, so you deserve the truth. This is simple. This can be taught at No Name U. Now, you were taught Nirenberg's and Mattei's experiments where they took ooh and they got for a reason. The most common genetic mutation in human beings affects precisely that codon. Cystic fibrosis, which kills all people affected with it by age 40, is caused by the loss of one codon, TTT, in the DNA encoding the CFTR gene. It's a deletion of 3 base pairs. Is that also a missense mutation? If you want to call it that, go ahead. All geneticists would call it a deletion. Is the protein still produced? Yes. Why is the disease-causing allele recessive to the wild-type one? Because the disease-causing allele produces a protein that cannot fold properly. And instead of sorting to the cell surface, it is stuck in the endoplasmic reticulum. And why am I telling you this? I mentioned to you that there's a company called Vertex, which built a drug which rescues the function of mutant CFTR. And that drug has shown efficacy in alleviating the symptoms of patients with cystic fibrosis like nobody has ever seen before. But I also mentioned to you that the drug they made is against a less common allele. This means that the most common allele of CFTR that causes the disease causes a disease that remains uncurable. And this is a challenge for your generation to try to address. How do we build a drug that corrects the misfolding defect of the most common allele of CFTR that causes CF? Okay, we discussed nonsense mutations, and we discussed a type of a missense mutation which deletes one codon, one amino acid is lost. But what is similar in both cases is the mutant allele is completely and fully recessive to the wild-type one. So the parents of children with cystic fibrosis have themselves no lung or pancreas pathology whatsoever. And now it's going to get much more elaborate. I think I spoke about this gentleman. His name is Ryan Clark. He plays for the Pittsburgh Steelers. Okay, Steelers. He has sickle cell trait. And sickle cell trait is, first of all, a condition that's quite prevalent in the United States. An even more severe related condition is also quite prevalent. 80,000 African Americans are affected with sickle cell anemia. And Miles Davis was an example. Episodes of acute pain, lung susceptibility to lung infection, and severe susceptibility to stroke. Now, you should read why Ryan Clark why his coach and Ryan himself chose to not have him play at Mile High Stadium. I'm telling you this because this highlights to us the challenge of ascribing simple claims of dominance and recessivity to alleles. So the allele of beta globin that causes sickle cell anemia is a classic missense mutation. One point mutation, one change in one codon, one substitution of one amino acid for a different one. The wild type beta globin chain contains a glutamate. The sickle cell anemia one has a valine. So it's a glutamate to valine substitution. People homozygous for the wild type allele have no symptoms related to that constellation of traits. People homozygous for the recessive allele have all-out severe disease. It's a very severe condition, sickle cell anemia. People who are heterozygous are actually fine. I mean, look at this gentleman. He's fit enough to play one of the most physically demanding athletic endeavors, known to human beings. He's a professional football player. That's quite something. It is a missense mutation, and if a patient of yours says, well, I've heard that missense mutations are recessive or dominant, you need to know the nuance. And in this specific situation, that specific mutation creates an allele that's recessive to the wild type one, but incompletely. So this gentleman has milder symptoms, much more mild. I mean, it's not nothing, but they're much more mild than patients with all-out sickle cell anemia. Furthermore, and here's an important point that I'd like you to remember for the rest of your life. He can play just fine in his hometown of Pittsburgh or anywhere else where the National Football League plays. He just doesn't want to play in Denver. So here's a situation where the environment modifies the presentation of an individual, or if we were talking about a model organism, we would talk about a phenotype. The environment modifies the extent to which an individual's genetic background creates that individual's physical makeup. And this gentleman is a prime and wonderful example. I actually taught you another, and confusingly, but it's just a fact, geneticists will switch between incompletely recessive and partly recessive incomplete dominance like this, confusing students, but I'm hopeful not confusing you. I don't suppose by some miracle any of you know who this dashing-looking, sort of romantically somber young man is. Wow! Oh, I am so impressed. It's Ernest Hemingway. As a young man, when he, of course, then went to Paris, he wrote, this is him as a soldier during the war. Of course, A Farewell to Arms comes from that. And then, of course, he went to Paris and wrote The Sun Also Rises. Just truly some of the most marvelous works of 20th century fiction. He then became an alcoholic and shot himself. "10,000 years ago, when folks in southeastern China domesticated rice, an allele of alcohol dehydrogenase arose, which prevents them from consuming any measurable amount of alcohol because their allele of alcohol dehydrogenase is much faster at metabolizing ethanol to acetaldehyde, and thus it gets so toxic to them they just don't even start. The allele of alcohol dehydrogenase that evolved in southeast China and then spread through mainland China and other parts of Asia is also a missense mutation. One amino acid has been changed into another. Now you can think about the inheritance of alcohol, of high rates of alcohol consumption as a purely dominant trait. So if somebody from Southeast China chooses to have a child and that child gets one allele of that gene from mom or dad, and the other allele is the European variety, that individual will also be insensitive, will be incapable of consuming large amounts of alcohol. However, the appearance of that individual will be intermediate between all-out inability to even approach a glass of beer at the one extreme and drinking oneself to death at the other extreme. So I just walked you through two examples of missense alleles. And one thing you will note is I haven't yet spoken about, quote, function, unquote. We talked about the fact that the allele of, let's see, which example did I use for, the allele of cystic fibrosis that causes, CFTR that causes cystic fibrosis is fully loss of function. The protein no longer functions. The allele of SCNNA that causes insensitivity to pain, fully loss of function. This alcohol dehydrogenase allele that metabolizes alcohol faster is what? Gain of function. Thank you, Maxine. Thank you, Katerina. Good for you. It acquires the ability to do something that the original allele does not. Here's an example in which geneticists see a gain of function allele, and that gain of function allele is dominant. Why? Well, because in a heterozygous individual, the wild-type allele sits there and does its thing, and the dominant allele, the gain of function one, has acquired an ability to do something else. And that ability predominates in the phenotype. Is there finer points of nuance here with respect to comparing how geneticists talk about incomplete dominance for sickle cell anemia and why they call the beta globin, this allele that causes sickle, fully recessive, and at the other extreme, geneticists call the allele of alcohol dehydrogenase that metabolizes alcohol faster dominant, but both of them are missense mutations? Yeah. But by now I've spoken about this enough where I know you have the complete sort of stack of nuance in your head. your textbook contains a schematic that describes the various kinds of mutations that humans can get, but does not contain, I'm sad to say, the mutation that causes Huntington's disease. In other words, a mutation that's purely binarily dominant. And that's a shame. And I didn't teach you that for that reason, because I couldn't assign you reading from the book. But I need to tell you very briefly about this, because I will be able to inform you, I'm hopeful, of several things. First, let me introduce you to another database that you will find useful in your graduate career, your medical career, your public health career, and in your life as a human being. Online Mendelian Inheritance in Man, OMIM. Just Google the word, click on the link. OMIM is a manually curated, typed by hand by a trained professional, database that collates all available information about human genetic variation, in particular, human genetic variation that relates to disease, importantly, that is transmitted through generations in a Mendelian pattern. Go to OMIM and look up Huntington. It's the gene mutations in which cause Huntington's disease. Before you do that, I encourage you with all my heart to do two things. First, go to this URL and read an article by Amy Harmon about this woman, Catherine Moser. I met her. She's marvelous. But she knows that 4 or 5 years from now, she will start developing a completely devastating constellation of neurological disorders that will progressively get worse. And about 15 years from now, she will die. And most people with Huntington's disease die because they choke to death. And there's nothing they can do about them. Why does she know this? Well, because the disease runs in her family, and the disease is dominant. Namely, an individual heterozygous for the wild type and the disease-causing allele will develop the disease. I encourage you with all my heart to read this article, read about her family, and also read about Woody Guthrie, the man who wrote This Land Is Your Land, the founder, to all intents and purposes, of American popular music of the 20th century. He died of Huntington's disease. He spent the last decade of his life in completely devastating circumstances because nothing could be done back then, and unfortunately, tragically, to this day, physicians can do absolutely nothing to prevent the spread, to prevent this disease from killing people. We actually work on this quite hard, but I don't want to talk about this. Instead, I want to talk very briefly about the allele of Huntington that causes this disease. The allele is a triplet expansion. So in that regard, and this is the other reason I taught you about CODIS and short tandem repeats, the allele of Huntington that causes this devastating disease is an expansion of a repeat in the gene that encodes this protein. It is dominant because the mutant protein that has this expanded number of amino acids where there formerly were a short number is toxic, and it kills the neurons, and the patients develop pretty devastating symptoms. Will this be on the midterm? No. I didn't teach you this in a setting where you would have enough time to study this. Do I encourage you with all my heart to read the article and study this on your own time? Absolutely. I wanted to spend at least 5 minutes walking you through an issue that was raised by your GSI, Julia, who I know is in Evans, proctoring the students who are taking the clicker, was there. Hello, Julia. Hello, those in Evans. Mutations in BRCA1 that cause a hugely increased risk in the likelihood of developing lethal breast and ovarian cancer are nonsense mutations. Not all of them, but many of them are. So a DNA base is changed. That creates a stop codon prematurely in the messenger RNA. A truncated protein is produced. In fact, no protein is produced. So what is that allele? Loss of function or gain of function? Loss of function, absolutely. We just discussed an example of a condition in which, in fact, two, in which loss of function alleles thus are inherited in autosomal recessive fashion. That, however, is completely inconsistent with what I taught you about this condition. We know that Mrs. Lindner is a heterozygous for that allele. One allele of her BRCA1 gene is wild type, the other allele of her BRCA1 gene is mutant. She developed breast cancer and had to have major surgery. Furthermore, her daughter, Dr. Lindner, is also heterozygous for one wild type and one mutant allele, and she knows with 100% certainty that she has an 80% chance of developing lethal breast cancer, and she chose to have surgery to prevent that from happening. I am describing to you a genetic paradox, a disease, I beg your pardon, a condition caused by a loss of function allele, a nonsense allele that appears to be inherited in dominant fashion. What's going on? Let me explain this to you. I didn't discover until I prepared this lecture where the three bears came from, not the race around San Pablo Dam Reservoir, the original fairy tale. I thought it was a folk story. It wasn't. It was written by a romantic poet named Robert Southey. The Goldilocks effect, not too warm, not too cold, but just right, also applies to genes and is known as the effect of gene dosage. What's happening in the case of BRCA1 is one of the many, many examples where gene dosage comes into play. I'm going to read you two sentences from a paper that just came out about this. In fact, the story became more or less clear completely only in the past 5 years or so. Biallelic inactivation of the cancer susceptibility gene BRCA1 leads to breast and ovarian carcinogenesis. Paradoxically, BRCA1 deficiency in mice results in early embryonic lethality. Translation: if you remove that gene, you cannot get a mouse. Similarly, lack of BRCA1 in human somatic cells is thought to result in cellular lethality in view of BRCA1's essential function. How is it then the case that inactivation of BRCA1 in a woman leads to breast cancer if that protein is essential for cell function? What's going on? You saw this diagram on Monday in Dr. Maughan's lecture, and I need you to know what Dr. Maughan said about this and what your textbook says. I want to add one additional point of nuance. First, panel A shows that BRCA1 is a tumor suppressor. It is. Loss of function mutations in tumor suppressors lead to cancer and, in fact, are required for cancer. Both alleles of the tumor suppressor gene must be inactivated by loss of function mutations before cancer can begin. Now, unfortunately, this states that two independent mutations of the BRCA1 tumor suppressor occur in sporadic breast cancer. I'm sorry to say, and please make a note of it, this is just plain wrong. Mutations of BRCA1 and BRCA2 are never found in sporadic breast cancer, which represents 95% of its cases. So this is just the folks who wrote your textbook, which is very good, simply didn't fact check carefully enough. That said, you should still learn from this figure that key tumor suppressor genes, such as P53, which Dr. Maughan spoke about, and retinoblastoma, the retinoblastoma protein, do undergo this process. Both alleles need to be inactivated by loss of function mutations for cancer to proceed. And now a discussion, finally, of why Deborah Lindner, despite being heterozygous, she has one wild-type copy. The other is a loss of function. Why is she at risk? Because the BRCA1 protein is a guardian of the genome. Specifically, it prevents against mutation. 2 alleles worth of BRCA1 protein are required to protect the entire genome. 2 alleles worth of the BRCA1 protein are required to protect the entire genome. In an individual who has 1 wild type and 1 loss of function allele, there is half as much protein. And that is not enough. This is an example of a gene dosage effect. So in a person who is a carrier for this mutation, the genome is prone to mutation more so than an individual homozygous wild type for that gene. As a consequence, mutations occur throughout the genome at a heightened frequency than in individuals homozygous wild type. And the likelihood of a spontaneous mutation destroying or inactivating the other allele of the BRCA1 gene is higher. Paz is nodding. Did I explain that okay? Andrea, Maxine, Katerina, folks? Okay. That explains why the disease, while being caused by, I beg your pardon, the susceptibility to disease, while being caused by a loss of function allele, is inherited in a dominant fashion. It's a gene dosage effect. One more thing. This will not be on the midterm, but this will be in your clinical practice and graduate school. I showed this slide in introducing Dr. Mond. Heart disease, cancer, asthma and emphysema, stroke, Alzheimer's and diabetes. 1, 2, 3, 4, 5, 6. 6 of the 7 leading killers in our country today are largely not caused by Mendelian processes. Single mutations in single genes with enormously strong effects. In fact, the majority, the overwhelming majority of cases of all of those conditions are caused by non-Mendelian processes. Some of you might feel that you've been cheated out of your tuition. You worked so hard to get into Berkeley. You paid good money, or your parents did, or some combination thereof, to help you survive here. And you've just spent 12 lectures listening about stuff that I've just told you is not directly relevant to human disease. It is. Mendelian processes and Mendelian genetics provides a foundation for understanding all of biology. It just so happens that on planet Earth today, heart disease is not Mendelian, nor is susceptibility to most cancers, nor susceptibility to diabetes, nor emphysema. It's just a non-Mendelian process. And the reason I didn't teach you this is because nobody understands what's going on. We understand what to do, but we just don't understand what's happening. For example, 90% of variants that predispose to heart disease, Alzheimer's, or asthma are in fact non-coding. Why am I showing you a picture of Hugh Jackman in extremely silly makeup? Any guess? What non-coding mutation did we discuss? Thank you. Good for you, Nicole. Lactase. The mutation that causes lactase persistence is a mutation in an enhancer of a gene. It's not coding. It's neither a silence, nor a nonsense, nor a missense, nor none of that. It's a mutation in an enhancer. It doesn't cause an effect on protein structure or sequence. It causes an effect on the regulation of genes. And figuring that out is a challenge for your generation, which is why, and we don't have good models yet, which is why I'm not teaching you this. That said, since I have 14 minutes, and with that, you are free to go, and we're done. I wanted to first reward you with a clicker quiz. While you're getting ready, the clicker quiz, as promised, is about the BRCA1 figure. Dr. Lindner, shown here, has a 50% chance of inheriting the loss of function allele of BRCA1 from her mom, Mrs. Faith Lindner. Why is that the case? A, well, humans are diploid, and the above is true for all autosomal loci, and BRCA1 happens to be autosomal. Or B, homolog pairing at the metaphase plate of meiosis 1, followed by their segregation into distinct daughter cells. C, Mendel's first law. D, Mendel's law of equal segregation. Or E, any and all of the above. With that, pull out your clicker, and let's take a very short length of time to answer that question. Let's take 60 seconds. As always, Google, etc.
By the way, Professor, I accept your apology about the banana. We're good.
Let's do 10 more seconds. 7, 6, 5, 4, something or other. 1. All right. And of course, this is the last clicker quiz. And so in the spirit of, there is no wrong answer. All answers are correct. But everybody gets a point. But yes, of course, thank you. E is the correctedest. But that said, everybody gets a point. No, wait. Wait, stop. I'm not done yet. I have 11 more minutes. Go away. All right. Okay. Put away, if you want to, if you need to go, go. You're not going to hurt my feelings. I want to teach you about epigenetics. I will speak at my normal speaking speed, not at my Bio1A sort of iTunes-friendly speaking speed. It's what I did my PhD on. It's what a whole bazillion people work on. It's completely next-gen. It won't be on the midterm, but this is what everybody who works on this works on. What's epigenetics? Well, genetics is when you have a mutation in a gene, and that change in a gene causes a change in phenotype. Epigenetics is a meiotically or mitotically inheritance of a phenotypic difference that occurs without a change in DNA sequence. So that patch of fur and that patch of fur in a calico cat and a female calico cat, that's essential, have the exact same DNA sequence, but they have a different phenotype. Why? Because of an epigenetic difference. Beautiful example of that is vernalization. Persephone, of course, in Greek myth was stolen, dragged down by the, I forget the deity of hell, Hades. Very good. And then the parents pleaded with Hades to release Persephone once so that she could talk to the parents, say, hi, mom, hi, dad, back to hell. And that, of course, is when spring came to Earth. Why am I telling you this? Because most plants in climates other than such as Northern California, where it never snows, go through vernalization. It's a process through which prolonged exposure to cold promotes flowering. Monocarpic species, writes Ricamassino, senesce after flowering and setting seed. But plants that require vernalization to flower thus typically require 2 seasons to complete the life cycle and are classified as biennials or winter annuals. Many winter annuals and biennials become established in the fall, taking advantage of the cool and moist conditions optimal for their growth. The vernalization requirement of such plants prevents flowering until spring has actually arrived. So in the spring, the plants remember that they were once frozen, but they are no longer frozen. They are already thawed. How do they remember? Epigenetic memory. So here's a normal species of brassica, the cabbage, which has gone through normal vernalization. And if you just keep it under warm conditions, it didn't know that winter has come, so it doesn't know to start flowering and senescing. It just keeps growing. it is reasonable to refer to the vernalization-induced mitotically stable, so it's stable through cell division. A plant under snow thaws and grows, but it remembers it was once frozen and it needs to start flowering. If it was never frozen, it will never flower. The competence to flower is an epigenetic switch. It is a change that can be propagated through cell divisions in the absence of the inducing signal. This is an expression state that propagates through mitosis, so through the life of an organism. Plants have epigenetic states that propagate through meiosis, so that propagate through generations. ZMAs, the second most important crop on Earth after only rice. I got these data from Jay Hollick, a professor here in UC Berkeley. This is the tassel, the male flower of the corn plant, and it comes in different colors. If you take a pure line of green corn plants and cross it to a pure line of purple, all the progeny are green. So green is completely dominant over purple, and you do a test cross, and all the progeny are green. And where has the purple allele gone? Boarded a ship? This is completely non-Mendelian. You should get a 1-to-1 ratio of purple to green in this test cross. You don't. You get all green. This is paramutation. Read Professor Hollick's review. The green allele shut down the purple one. The sequence is there, it's just no longer expressed. And that shutdown state is meiotically stable. And that silencing persists through meiosis. So it's called paramutation. It's not really mutation. The DNA is still there. Has there been a change in DNA sequence? No. Is the allele expressed? No. What is the difference in the DNA sequence between that and that? There is no difference in DNA sequence. Yes? Thank you for saying so. They are genetically heterozygous, but they're phenotypically homozygous. Because the silenced allele has silenced the active allele. Now, it's like the Borg or the Daleks. Exterminate, you will be assimilated. This is run on an epigenetic mark called DNA methylation. In the human genome, DNA methylation occurs within the dinucleotide CG, and it is thus symmetric. So one occurs on the Watson, one on the Crick strand. We have a machine called DNA methyltransferase that after DNA polymerase replicates a methylated piece of DNA, and thus this is hemi-methylated. Methyltransferase runs through the nucleus and says, remethylate, exterminate. You will be assimilated. You saw these gorgeous drawings from Dr. Mond on Monday. She didn't tell you because, just out of humility, I guess. She actually drew all of this herself. This is her own artwork. I know. No, it's remarkable. That the progression from this to this via this and is actually driven by this biallelic loss of tumor suppressor function in prostate cancer. Is not a deletion, is not a frame shift, is not a missense. It's epigenetic silencing. So the tumor suppressor genes in the transition from this to this are still there. They just have a molecular gag order. And small molecules that reactivate the silencing are all the rage in the field of cancer therapeutics. A representative example, look up in PubMed, enhancer of zest. To explain to you why you should look up enhancer of zest, I need to tell you one experiment that was done by Steve Jacobson at UCLA. He works on the flower in Arabidopsis. No comment on the nomenclature. He found a flower which has twice the number of male reproductive organs. And he called that gene Superman. I'm not responsible for the nomenclature. So here's a wild-type flower, and here's a Superman loss-of-function flower. It then gets even more entertaining, believe me, however hard may this be to conceptualize. There's two ways to mutate the Superman gene and get the same phenotype. Just delete it. Or you can silence it by methylation. So he decided to call that methylated form an epi-allele of the Superman gene, and it is called Clark Kent. Not making this up. He then did a screen. This is wild type, and this is Clark Kent. He then did a screen for things that are required for the maintenance of Clark Kent. What does a screen mean? Exactly what Beadle and Tatum did. He did a forward genetic screen for genes that would suppress Clark Kentitude. And he found a gene that does suppress that transition, which of course he called kryptonite. So kryptonite suppresses the Clark, I'm not making this up, kryptonite suppresses the Clark-Kent Superman transition. Kryptonite is a histone methyltransferase. So it methylates the tails of the proteins that pack the DNA. So what Steve Jacobson showed is kryptonite methylates the histones, and that methylated histone epigenetic mark recruits DNA methylation, and that silences Superman completely. Enhancer of zest, the aberrant activity of which causes metastatic prostate cancer, functions via the same mechanism. Thus, this is my last data slide, then there's one poem and you're done. Thus, you think about replication of DNA as propagating only genetic information. No. Epigenetic processes provide a mechanism by which our genes learn from experience. And epigenetic marks, whether on DNA or on histones, spread through replication and sometimes even through meiosis. I cannot do justice to this in the amount of time I have, in part because much of this is not understood. But if you're interested in getting started on learning about this, take an upper division genetics class, and look up papers from Jasper Ryan here at Berkeley, or Dave Alice at Rockefeller. All right, one slide of parting thoughts, one poem, and then we're done. Office hours, right after class, 180-10. If you find yourself too tired to study for the midterm, watch an episode of QI. Go to YouTube, type QI, and watch it. Trust me, you will. Just make sure that your parents are not in the room. Or sort of small children. Do not decide on a career in experimental science without working in a research lab first. Fortunately, you are students at one of the top, if not the top research university in the world. But don't go to grad school without working in a lab a little bit. If you have any questions, you can always write me an email. Finally, this is William Wordsworth, another romantic poet. And at the end of, I guess, the 18th century, he wrote this glorious poem, which I'll read to you quickly, and then you're free to go, which was anti-science, more or less, and pro-romantic view of life. He writes, "Up, up, my friend, and clear your looks. Why all this toil and trouble? Up, up, my friend, and quit your books, or sure you'll grow double. Books, 'tis a dull and endless strife. Come, hear the woodland linnet." I looked up the woodland linnet. It's not very descript, but I guess it sings beautifully. "How sweet his music, on my life, there's more of wisdom in it. One impulse from a vernal wood may teach you more of man, of moral evil and of good, than all the sages can." And then he writes his most famous lines. It's very well known. "Sweet is the lore which nature brings. Our meddling intellect misshapes the beauteous forms of things we murder to dissect." Enough of science and of art, continues Wordsworth. Close up these barren leaves, come forth and bring with you a heart that watches and receives. I'm hopeful that what you are learning in this class is that the two are not incompatible. If you bring with you a heart that watches and receives, then science is the realm for you. Good luck on the midterm.